So then we're going to move on into the proximal convoluted tubule. Now, it's key to remember that along this whole length of tubules that we're really now dealing with reabsorption and secretion. And the proximal convoluted tubule, absolutely reabsorption is what dominates. In fact, your textbook says, we'll talk to about secretion when we get to the DCT, even though it does truly happen here in the proximal convoluted tubule as well. Now, because the PCT is so interested in reabsorption, we see this function show up in our anatomy, in our structure. So if we look at the proximal convoluted tubule, notice a lot of this tubule structure, we actually see simple cuboidal cells. So in the PCT, it is in fact cuboidal cells, right here in the proximal convoluted tubule, a simple cuboidal epithelium, and in this case, it actually has, you probably can't read that, microvilli. Right, so now let's see if we can zoom in. Right, so here they're showing you those same simple cuboidal cells and the microvilli. That's to increase the surface area and therefore increase our ability to do reabsorption. So of all the things we filter out at the glomerulus, something like 60 to 70% get reabsorbed just here in the PCT. And in fact, if you look at things like the organic molecules, the glucose, the amino acids, vitamins, things like that, it's about 99% that are reabsorbed here in the PCT. So when you think proximal convoluted tubule, you think reabsorption. Okay? Again, over half of what we squished out, filtered out, is reabsorbed here, and it is 99% of all those organic molecules. Again, things like glucose, amino acids, fatty acids, vitamins, right? Basically all of them. And think about it, these things are all expensive, right? Amino acids are expensive. Energy filled glucose molecules, we don't want to let those go in the urine, right? Of course, we're also reabsorbing things like water, ions, and so we'll look at this as well. So here, this looks really complicated, but notice what they're showing you. Every time they have a blue arrow, it is showing reabsorption. That should be super clear that this is molecules moving from the tubule fluid, from the filtrate, back through these simple cuboidal cells into what we call the paratubular fluid, that's just extracellular fluid, and then on into the capillary, into the blood supply. So reabsorption is filtrate going back to the blood. Now, what they start looking at is how this happens. So let's do a quick reminder of some of the processes where molecules would actually move here. Okay, because they're showing you all this counter-transport, co-transport, different exchange pumps. Okay, so remember when you look at your membranes of cells, they're a phospholipid bilayer studded with proteins, right? Lipid-soluble molecules are able to diffuse directly through that, so things like oxygen and carbon dioxide. Again, diffusion is a passive process, no energy required, because these molecules are moving down their concentration gradients, right? From an area of high concentration to an area of low concentration. Now, we have other substances that are going to diffuse as well, but if they're not lipid soluble, they are going to need some kind of protein to help them get through there, right? So things like water actually need a channel protein, small ions, but they still can use this passive diffusion where they move down their concentration gradients. And of course, we can also use active transport. So what I did want to show you is notice this example. They're talking about carrier proteins, and notice this is a form of facilitated diffusion. Because it's diffusion, we know two things. The ions, the molecules are moving down their concentration gradient, and then it's a passive process, no ATP required. The facilitated part is telling you that you have a protein helping. This is one that tends to throw students though, this idea of a carrier protein, because notice what happens here for glucose transport. Glucose actually binds to this carrier protein, and the binding of that molecule actually changes the shape of the protein and releases the glucose on the other side of the membrane. But this is passive. That one's confusing, right? But the protein, the shape change is really an interaction, right? Different bonding that's happening between the glucose and the protein, and it just is a conformational change. We're not actually pumping it against a gradient or anything like that. So this is passive, right? And this is how we see glucose coming into our cell. Actually, this one's using co-transport. Let's look at that. Okay, before we get there, the active transport, of course, all requires ATP. And this is our classic example, the sodium-potassium pump. Right, here we see 2 potassiums being brought into a cell and 3 sodiums being kicked out. Now, a couple things are your clue that this is active transport. This is the big one. It's utilizing ATP, it's using energy. But the other is that this is really a pump. It's working against a concentration gradient, right? Inside of the cell, we have lots of potassium, and this is bringing more potassium in. Right? There's lots of sodium outside of the cell, and we're pushing more sodium out. Right? So that is active transport. Now, this is a super cool one, right? So they're calling this secondary active transport. You'll see this in this chapter referred to as co-transport. So look what happens here. We have a carrier molecule. We have a situation where sodium wants to move down its concentration gradient, right? And sodium comes in and binds, so does glucose, and when that happens and our protein changes shape, we can actually, in this case, move glucose against its concentration gradient without technically using any ATP, because sodium is moving down its concentration gradient. So as long as one of these ions, one of these molecules is moving down its concentration gradient, we can kind of sneak the other one in, right? And so this is what they refer to as co-transport. Okay? And so here they're showing how this is really, truly reliant on the use of energy in our sodium-potassium pump to keep pushing that sodium out. But as long as there's more sodium out, then we can kind of freely sneak our glucose in. Okay, so what they're trying to show you here is at the PCT how all these different molecules, right, glucose, amino acids, water, ions, all these kind of things are moving in and out of the cell. Don't get too lost in the details here, right? I want you to realize that reabsorption is dominant, what's coming in, but this is kind of, I think this is pretty interesting, right? So here they're showing you in green a co-transporter. This is how glucose and these other organic molecules are actually getting back into the cell and then into the blood, right? They're sneaking in with sodium, and then they're able just to diffuse out to the blood. We also see, here's our sodium-potassium pump, right? Sodium getting kicked out, potassium being brought in. A lot of this has to do with matching up ions, right? So not getting like too electrically imbalanced, right? So as sodium's coming through, we might see something like bicarbonate coming with it. Where's our other one? So much sodium is coming this way, so things like potassium go in, hydrogen go in. Here we see sodium and hydrogen being exchanged. So a lot of times if you see one positive charge coming in, another positive charge needs to go out. And some of this will end up being important. We'll come back to thinking about when sodium is reabsorbed, brought into the blood, we're kicking potassium out of the cell. That'll be really important in some of our fluid electrolyte balance conversations, right? Or over here, you can see all our decisions about pH, right? So if hydrogen is being secreted right, being sent to the filtrate, that would help us bring our pH up towards neutral, right? And at the same time, you see that bicarbonate being reabsorbed, brought to the blood, right? So we can use those things together. Is hydrogen being secreted or reabsorbed? What about bicarbonate? Okay, and then of course water is following all these ions through osmosis. So we're going to see this basically as we go down the rest of that tubule as well. Another thing to think about. So all of these transport proteins, carrier-mediated transport, all of this, they can become saturated. So what we run into is what's called a transport maximum. This is basically the concentration of that substance, right, glucose, sodium, whatever it is, the concentration of the substance where the carrier protein gets saturated. Right, so it's much like an enzyme in that way, right? And so it's kind of like, you know, if someone was handing me markers and I'm handing them off, right? It's only so quickly I can do that. At a certain point I get saturated. I can only move so much of a substance, right? And so that is called the transport maximum. So as this filtrate is coming by the proximal convoluted tubule, if we hit this transport maximum, if, say, this glucose transporter is saturated, it's not going to be able to keep bringing glucose in. That glucose will stay in the filtrate and eventually show up in the urine. And so we call that level the renal threshold. Right, basically where this starts to, the concentration, where that substance appears in the urine, right? It's not getting reabsorbed. And these two definitely go together. The transport maximum is the level where that protein is saturated, and that leads to this renal threshold. It's going to start showing up in the urine, right? So in lab, you'll actually do these you'll do urine test strips. Glucose should not be in your urine. If glucose is in your urine, that means there was so much glucose in your blood that was filtered, right, at the glomerulus. So much of it ended up in the filtrate that when it passed the PCT, you maxed out those enzymes and it had to carry on, and it shows up in your urine. So urine can actually be really instructive in a few different ways. Two other things before we leave the PCT. Do keep in mind that while reabsorption is dominant, secretion takes place here as well. In fact, we see it in this picture, right, with the red arrows. In this case, they're showing hydrogen being secreted. Remember, secrete it to excrete it. This is something we're adding to the filtrate to eventually become urine. So here, for example, we see acids being added. There is some limited secretion of different drugs, some toxins, right? Things like ammonia can be added in here. I know creatinine, for example, so some different metabolic byproducts, right? Anyway, the big picture is there is some secretion at the PCT. The other thing that I wanted to mention then is as we're moving on, all right, there's my clicker, we're about to move on to the nephron loop here, right? And as we're in this attempt to make urine, right, by the time we're over in the collecting duct, a lot of the time we're going to follow what's called osmolarity. And so if you see them start talking about osmolarity in this chapter, this is essentially the saltiness of a fluid, right? So it's the concentration of the salts in this fluid. Your blood is about 300 milliosmoles. Again, this is the number of solute of salt particles in a liter of water. This is pretty dilute. So for comparison, fresh water is about 5 milliosmol, right? So really dilute, but there's a little bit of salt in there. If we look at ocean water, it's about 1,000 milliosmol. So when we took the blood and we filtered it, right, we squished out a bunch of water and some other salts and stuff, we made a really dilute, maybe more like fresh water. The filtrate was very dilute. By the time we're done reabsorbing things here in the PCT, at the end of PCT, we're right back to about 300 milliosmol, right? We brought a lot of those salts back in. But here's the thing, if you want to make a concentrated urine, we haven't done that yet. So realize that's going to happen in the rest of the tubules. For humans, about the most concentrated urine we can make is about 1,200 milliosmol, which is why you really can't drink ocean water, right? They talk about that dehydrating you even more because it's just so salty, and our kidneys can't really concentrate much higher than that water. Okay, so we're going to head off to the nephron loop.